Good evening, everyone. As said, I'm talking today about work I completed during my PhD looking at enzymatic blood group conversion. So to understand this, we need to understand a bit of the background of blood groups. So this is what you'll know if you ever have given blood or had certain medical procedures. It's the AB or O or AB blood type that you'll have been told. And this was defined based on red blood cells. And on the surface of red blood cells, you have markers or antigens, which can be type A or type B. If you're blood group O, you don't have either of these markers on the surface of your cell, and if you're blood group AB, you have both. Now, despite these being blood groups, these markers are actually on lots of different cells in the body, and importantly, they're on the surface of your blood vessels. So if we have a look at the actual vasculature itself, we can imagine it as the series of tubes within the body, and if we split them transversely like that, you can see the sort of cross-section of what we're going to be talking about mainly, which are the capillaries, and those markers are expressed on the inner surface. Now, my work is all about the human kidney, so here is actually an example of a blood group B kidney, and we can see that the magenta color on the left-hand panel, that's the blood group B antigen in all of these circles. CD31 is a marker of blood vessels, and we can see that they co-localize really nicely. Throughout the talk, you'll see pictures that look a lot like this, and this is demonstrating that cross-section of a capillary, where we can see those markers expressed on the inside. So for those chemists amongst us who are interested in this, this is the proper structure of these markers. They're blood group specific, and they all start with something called the H antigen. All of this is to do with sugar groups bonded together. So the H antigen are these three sugars, and that's on every single blood group, including blood group O. It's actually the terminal sugar, which in blood group A is this, is the blue square, and in blood group B is the yellow circle. So that's what actually makes you either an A or a blood group B marker. Now, for those who are less chemistry inclined, the H antigen is a flagpole on every cell, and type A people show an A flag, and B people show a B flag. So, why does this matter? Well, every person expresses antibodies against the type of marker you don't express. So this happens natively, and in type O individuals, this means you'd raise anti-A and anti-B antibodies. These are circulating around in your blood. For type A individuals, you'd raise anti-B antibodies, and vice versa. And if you've got both of those markers on the surface of your cell, then you don't actually produce any circulating antibodies against them. This causes problems in transplantation because you're taking an organ from one individual, which may express some of these markers with those flags, and if they interact with circulating antibodies, that can cause all sorts of problems. So what would actually happen? A blood group A kidney going into a blood group B patient would involve a lot of A flags on the kidney interacting with the anti-A antibodies inside the recipient. Those antibodies would bind immediately and start causing rejection responses instantly. You would see this within minutes of surgery. So we end up with compatibility restrictions. So type AB recipients can receive from everybody because you need to match those markers. Things get more restricted as we go through the others. So blood group A people can only accept from type O and B because they can't interact with any of the type B markers. And vice versa for type B, they can't have anything with an A marker. Blood group O is actually the most restrictive. You can only actually receive from another blood group O donor. And this brings us on to the problem in transplantation, which is a lack of equality in the access to compatible organs. Here's a graph showing the waiting time for a kidney transplant in the UK. This is the most up-to-date data as of 2023. And we can see that for blood group O and B, you're waiting 2 to 3 times as long as the other blood groups. And this is purely based on the compatibility restrictions faced by these groups alongside higher precedence of kidney disease amongst blood group B individuals. So a possible solution is enzymatic blood group conversion. So enzymes work as biological catalysts, effectively molecular scissors. And what they can do in the case of blood group A is a class of enzyme called azymes, nice and neatly, can literally use some scissors and chop off the A flag and convert it back to the flagpole or the H antigen. A class of enzymes which we've called B-zymes do the same with the type B. So what this means is you could take either blood group antigen and convert it back to the universal state through the addition of these molecular scissors. This was applied in my PhD to whole human kidneys, so I'm just going to warn people that there are images of human kidneys on the next two slides. I'll say when there are no more images available, in case people would like to look away. So this involved taking human kidneys declined for transplantation and offered for research, and we put perfusate around them. It's an adapted sort of heart-lung bypass machine that can literally pump blood around a kidney outside the body. And this is at normal body temperature or whatever temperature we define. And here is a pair of kidneys. The right one is pink and well perfused, and this is a little video. You're about to see the clamp be released on the left kidney, and you can see that it will start filling with blood, and you'll see that it will pink up and become actually much bigger in size. And so this is happening experimentally within our lab. Now, the benefit of this is that you're able to directly target a human kidney and assess the effects of our treatment. So there are no more images of any kidneys from now on. My PhD looked at taking blood group A or B kidneys, adding these enzymes during whole organ perfusion in order to generate universal kidneys. So here is some data from some warm perfusion. So this involved body temperature, 37 degrees C, and I took pairs of human kidneys. This is a great experimental model because they're exactly matched for the donor sex, age, and comorbidities and past medical history, things like that. So in the control kidney, which is on the left-hand panel, you can see this lovely magenta color. That's the blood group A antigen. And throughout the whole of the pre-treatment 1 hour and 6 hours of perfusion, it doesn't change. We didn't do anything. So that was exactly as expected. In the treated column on the right-hand side, we can see that after 1 hour, it's turned green. The green color is the H antigen, the flagpole. So the pink flag has been removed, and now we're just left with the universal structure. And it happened as quickly as 1 hour. So then we thought, okay, if it works warm, can it work cold? And this matters because there are devices for cold perfusion, what's called hypothermic, which are completely transportable and battery-powered. And these are used commercially to date. The normothermic devices, the ones that do it warm, they're currently more experimental, and the actual devices don't exist commercially. These cold perfusion devices were used again with pairs of kidneys, and we can see in the control kidney, we've got a pre-treatment 1 hour, 2 hour, and 6 hours, and it stays completely magenta the whole way. And in the treated kidneys, that after 2 hours, it's turned green. So it's an hour slower, but it worked. And this was excellent for the concept of how we would move this into clinical practice. A big question, though, is, well, what happens if we put this into somebody? Because what matters is that antibody interaction immediately. So this involved taking the kidneys that were blood group A, and we converted them back to the blood group O status, and then I added antibody into the circulation to see what would happen. So in this panel, we can see that the yellow color is the added antibody afterwards, and after the treatment, but before the antibody, it's completely black in both, and then after we add the antibody in the control kidney, it's all gone yellow. This is because this control kidney had nothing done to it. It's got lots of A markers, and all the antibody has bound it immediately. In the treated kidney, those A flags have been removed. There is nothing for the antibody to bind, and it stays completely black. This is also shown in our circulating antibody data. So if you look at the red squares, we can see that that level of antibody didn't change over the 4 hours that we perfused the kidneys for. And in the control kidney, which are the blue circles, we can see the amount of antibody is slowly dropping out of circulation, and that's because it's binding the kidney. So the treated kidney is acting exactly like a universal kidney. It's not binding any of the antibody. The last aspect of this work that I'll be talking to you today is about the complement cascade. So this is part of frontline immune defense against foreign material. It's part of the innate immune system, and it locally triggers inflammation and blood clots alongside immune cell recruitment. There's this horrendously large schematic here. Please don't look in too much detail. But the point being is it's a long old proteolytic cascade, meaning that basically loads of proteins get cleaved into different components that have a biological effect. But there are only 3 I actually care about, so let me show you through those. The first is C1q. This is the one that actually starts this cascade, which would trigger all of these rejection responses. And in this panel here, if you look at the gray circle here, we can see in the control kidney that there is C1q. The yellow shows that it's co-localizing with where the antibody is, exactly as expected. And in the treated kidney, it's not there. It's not in this circle there. The next component is a split product of C4b called C4d. And again, it's only found in the control kidney where the antibody's bound. Our treated kidneys evade that response. And the last aspect is the membrane attack complex, which is the final part of this cascade, which causes cell lysis, so breakdown of the cells. And again, it's only present in the control kidney. Our treated kidneys can evade that hyperacute response. So the work so far has shown that these enzymes can be used to remove blood group antigens from whole human kidneys, and it's the first ever proof of principle work for this done in kidneys worldwide. We've shown it's clinically translatable. The short action times of 1 to 2 hours, depending on which device and which temperature, and using technology that's already approved for clinical use means that translating this should be fairly simple. And this has the potential to revolutionize the allocation of donor kidneys, directly impacting those patient waiting times. The dream being that we could take any kidney and allocate it to the next person on the waiting list, regardless of their blood group. So, the future is looking towards universal donor kidneys, the hope being any incompatible kidney can be turned into a compatible one and given to the next individual, removing the effects of a genetic lottery. I'd like to acknowledge all the people who helped with this work, but especially the donors and their families, without whom this research couldn't have happened. Thank you very much.
Thank you very much. You packed a huge amount of information into a short period of time and got to a wonderful conclusion. I'm sure we can generate some questions.
Fascinating lecture, thank you very much. Is it applicable to other organs for donation, which is the obvious question, I guess?
Absolutely. So this work is applicable to any solid organ for transplantation. So the most common, or the place where you'd have the most visceral impact immediately is kidney transplantation, just due to the sheer number that are happening worldwide. But the potential for using it in liver, heart, lung, etc. is definitely there. It's just the sheer numbers of those transplants are much less and they're much more difficult to match, whereas kidneys, we actually probably have a much better way to match these organs. We're just not doing it yet.
Thank you, that was terrific. This enzymatic conversion, is that applicable to any other organs?
So, as I was just touching on, this is actually only, the blood group antigens themselves are present on lots of different types of tissue. So we can find it in the heart. You actually don't find it so much in the liver. They're less immunogenic organs, so they don't worry too much about the blood group matching there. But the kidney is a huge one. You get that wrong, and sometimes they do. Typically, it's a clerical error. So somebody's written down the wrong blood group, which is not as disastrous as if it was a heart, which has happened before. But the kidney, you get the wrong blood group, and it will go black within about 30 minutes. So this is a highly vascularized organ that is very applicable to this technique. Thankfully, with normothermic or any of the perfusion techniques, you're targeting where the blood will go, which is exactly where the problem is in transplant.
So I think we're all entirely comfortable until you talked about human error.
Sorry. Oh, dear.
Can I ask, so you do this sort of enzyme thing that chops off the marker to turn everything into sort of equivalent to type O.
Yep.
But doesn't the organ, when it sort of regenerates itself, produce what it always used to?
Yeah, so this is a really interesting question, and it's actually what the next phase of our work is looking at. This isn't a genetic treatment, so this is a surface level change of the blood group, but we're starting to do some work now looking at much longer perfusions. We've done a couple up to 24 hours, basically providing a window where that transplant will be accepted, and then any therapeutic intervention could happen potentially after that. There are a couple other mechanisms in play in the body that mean that we're not too worried if they do come back, but in general, there's a window where you might be able to intervene post-transplant, and just getting a transplant into somebody will change their whole outlook. It's so much better than waiting on the waiting list.
So, there's been quite a lot of news about pig organs, mainly livers, that have been genetically modified and have been transplanted a couple of times, I think, in the US with some success. Is this relevant here?
So that's the field of what we call xenotransplantation. So that's taking another species and transplanting it into humans, pig being sort of fairly close. there are a couple of concepts that are applicable here. So the genetic change is something we're going to be looking at as another part of our project, is whether we can change this at the genetic level. But the simplicity of our work is that we can provide a simple one-off treatment in a clinically approved device in a short action time that works with current transplant schemes in the UK and worldwide. And if we can do that first, it will just generate more transplants. And then in the future, 10, 20 years, when genetically treated organs are more commonplace, then we can replace that. But at least in the short term, this provides an easily viable option to get over this ABO barrier.
John, you...
I have a question about your fantastic talk, by the way. I've never heard immunology explained so clearly. Your experimental technique for getting the micrographs, I presume you have many control kidneys and many treated kidneys, and you wait 6 hours for kidney 1 and section it?
Yeah, so they're serially biopsied throughout. Okay. So as I said before, we're very fortunate that every now and then we get a pair of kidneys that are declined for transplantation. So we're able to do them exactly the same time, where one kidney from the same donor will be left for exactly the same conditions, and the other one will have that condition plus the treatment. So we are able to sort of as closely mirror whether it's safe and effective against a biologically matched control.
And these are fluorescence label images we're looking at?
Yes, so immunofluorescence.
Yep. Thank you. Any last question? One at the back left.
Well, this is wonderful news you've given us. How many years are we away from going on a climb to volume availability?
So with this particular technique, we are hoping to do the first clinical work, all going to plan, within probably 3 years, I think. So that's the timescale we're looking at the current work. That will be a short clinical trial, and then from then, if it works effectively, we're working with a research group that spun out and now formed a company to make these enzymes. So they're pushing very hard to get it FDA regulated. So the actual use of this should be very easy to implement clinically, and should have immediate impact for some individuals who are just really struggling to match with a kidney transplant, of which sadly there are quite a few.
Thank you very much. That was a wonderful talk.
Thank you very much.
And really positive news.
Thank you.
Thank you so much. Yes.